Hello and welcome everyone. My name is Eric Franzen of SemanticWeb.com. We would like to thank you for joining us today for this webinar, a joint production between Dataversity and SemanticWeb.com, with our speaker, Michel Dumontierl of the Stanford Center for Biomedical Informatics Research. Today, Michel will be discussing data-driven biomedical research with Semantic Web Technologies. Just a few quick points to get us started. Due to the large number of people that attend these sessions, you will be muted during the entire webinar. We will be collecting questions in the Q&A box in the bottom right-hand corner of your screen. As always, we will send a follow-up email within 2 business days containing links to the slides, the recording of this session, and any additional information on resources that may come up during the webinar. And now a few words about our speaker, Michel Dumontierl. Michel is an associate professor of medicine at Stanford University in the Stanford Center for Biomedical Informatics Research. His research focuses on methods to integrate large, heterogeneous clinical and biomedical data for discovery. His research interests include 1, developing novel therapeutics for rare and complex diseases, 2, elucidating the mechanism of drug-induced side effects, and 3, optimizing multi-drug therapies to minimize undesirable side effects. Now to get us started on today's main content, please allow me to introduce David Booth. David has been one of the primary organizers of the Yosemite project, and he's going to put today's webinar into the larger context of that project. Hi, David.
Hi, Eric. Thank you. The Yosemite project articulates a roadmap for achieving semantic interoperability of all structured healthcare information, using RDF as a universal information representation. So today's webinar by Michel Dumontierl is slightly different from the previous webinars in this series in that it is not focused on any one step of the Yosemite project roadmap. Instead, it demonstrates some of what can be done with healthcare data in combination with other relevant biomedical data once the steps in this roadmap have been achieved. After all, healthcare connects with everything else, and this is nowhere more apparent than in biomedical research. So the Yosemite project roadmap shows how we can get to this point of semantic interoperability, capturing the full richness and diversity of healthcare information on a broad scale. And I should note that the Yosemite project is a collaborative effort, and anyone who is interested is invited to participate in it. So, Michel, thank you very much for joining us today.
All right, thank you very much, Eric and David, for the introduction. This is, I think the Yosemite project is extremely promising. It is something that we have been asking for a long time, and I hope to begin to illustrate what it is that we can do when the data, when we make efforts to normalize the representation of information, and particularly using the semantic web stack. So in this talk, I'd like to discuss some of the issues around having hypothesis-driven science and reproducible science, our efforts to build out a linked data for the life sciences, our efforts to also construct semantic clinical data warehouses and to use that to start asking questions of biomedical interest, and obviously leading to much more integrated translational research where knowledge that we gain from the biomedical research can inform healthcare and vice versa, where healthcare observations can also inform biomedical research and some of the future directions that our group and others are hoping to take. So science really begins with the observation, and nowhere is that clearer than the healthcare domain, where physicians are routinely making observations along with other healthcare professionals about a patient and their conditions, about the quality of care just as much as the treatments themselves and the outcomes. And eventually, you know, what we see is that things don't always go as planned, and we start to ask questions as to why is that the case. And we begin to formulate hypotheses about what could be happening, but it's really only in the context of having some data or performing some kind of experiment that we can validate any prediction about what the hypothesis entails. And this is really key. We are at the point where we have so much information that we really need to make use of that information to help inform the science of medicine. So the problem, however, is that finding the evidence that would support or refute some kind of hypothesis is actually very, very difficult. Often we are faced with anecdotal description, even to the extent that any kind of systematic investigation is still kind of limited. It's scoped to a particular set of variables, and it's not necessarily clear whether or not it can translate into other settings. And yet, at the same time, we have so much information, so many people going after the same kinds of questions. Wouldn't it be nice if we can aggregate these kinds of data and use them to investigate hypotheses? Now, to do that is no trivial task, as anybody knows. And so what I think is so exciting about the Semantic Web effort is that basically it's a way to standardize the publishing, the sharing, the linking, and querying of information that is relevant to us, facts, the expert knowledge, and the services that are available to process that data. And I think what's interesting is that because we leverage web technology, it means that at a very simplistic level, it is a scalable enterprise. If somebody has data and they can format it appropriately, then they can do so independent of other people. Now, of course, as we know, some collaboration is required in order to ensure that it becomes easier to navigate these web of data and to do kind of meta-analyses about that. So we and others are building a monstrous network of what we call linked open data. And some of you are probably pretty familiar with this diagram. This is linked open data cloud diagram. This one recently updated in August 2014. And there's a variety of different domains that are represented in the linked open data network, from publications, social networking, geography and government, and media. And in the lower right-hand corner in pink is the life sciences domain. And so we are one of the major contributors of the life sciences data through our project called BioTRDF. What BioTRDF really tries to do is focus on trying to normalize the representation of biomedical data by taking it from its variety of different formats, tab files, XML files, relational databases, flat files with arbitrary syntaxes, and turning it all into RDF, one common syntax that we can share the data with. We can annotate it with respect to its provenance. We can compute statistics about the nature of the information that is contained therein. And we can use a common query language, SPARQL, to access that information and to extract out the information that we are particularly interested in. So BioTRDF really focuses much more on the data conversion than on the data supply, although it does make some effort to enable those, as you've probably seen in other presentations, where we have the identifiers for the data objects. They are dereferenceable. You can look them up and get information about them. We have public demonstrative triple stores where you can start to compose some queries, federated queries across the different data sets and create complex queries that way. And we also make the data available. So you can download it yourself, load it in your own triple store, and perform your own kinds of queries. So BioTRDF in its latest release has nearly actually 11 billion statements from about 35 different biomedical data sets. And we have partnerships with a number of different organizations to link out and between the data sets. And this is the substrate for the research that we do. Now, one of the challenges about the semantic web data is that even though it's in a common format, it is still highly heterogeneous with respect to its representation. And that makes actually even just querying the same kinds of data relatively difficult still. You basically have to write to each data set for information that you're interested in. And for now, one of the ways that we're handling that is through a project called HyQue. And HyQue, the idea is to test hypotheses that you may have by trying to link components of the hypotheses with data of interest. So on its own, it is a platform for knowledge discovery. It uses semantic web technologies to access the facts, the expert knowledge that's embedded in ontologies and semantic web services. It provides provenance for its analyses and enables a reproducible evaluation using both positive and negative findings. So that is to say, when you have a hypothesis, for instance, that a drug could be used to treat a particular disease, we want to see those facts that support or dispute this hypothesis by looking at various components. For instance, you would expect that the drug might target genes or proteins that are related to the disease in the very first place. And it could be that some people have done experiments to test this and have found that that not be the case. And so these negative findings could be used to potentially dispute your hypothesis. And so HyQue is capable of doing this, although there isn't very much data in this format. So HyQue uses a number of semantic web technologies. One, it uses the linked data from Bio30F as a key substrate. It uses ontologies that can be found in the BioPortal repository. These biomedical ontologies, there's now well over, I think there's about 400 of them. Of course, HyQue focuses on those that are specifically linked to data. And there's also components that we're trying to integrate now to enable HyQue to access semantic web services so that it can use computational or analytical services to answer the questions. And all of this uses SPIN, which is basically a language that builds on SPARQL to evaluate information. And so we use that to assess the facts that are being returned from the knowledge base and ultimately evaluate the hypothesis with respect to the components that it could find. So we have used this in a few different use cases. In the first case, we looked at one that was expressed by the FDA, where they were interested in, they're launching a drug safety program to detect toxicity of compounds, hopefully prior to approval, but certainly even after approval, then during monitoring, active monitoring. And so in a sort of landmark paper, one of the key individuals published a paper to sort of articulate the kinds of data that would be needed in order to do this sort of activity and to follow specifically these tyrosine kinase inhibitors that are used to treat cancer but have been linked to cardiotoxicity. So the question is, for future compounds that are aiming to treat cancer, can we also avoid cardiotoxicity and identify those factors that contribute to it? And what was really interesting is that one of the figures in their paper listed a set of data that could be used to inform the FDA and others about the clinical cardiotoxicity of compounds. And what I have here is in the arrows in red are those data that are already in Bio30F and then others and also generated by others. So for instance, Ractome and the Array Express archive are provided by the EBI in RDF. And so we have a very large covering of these data already. So then we ask the question, well, what is it about these data that you would like to have access to? What is the information that we need to gather and present to you in order to show you evidence of cardiotoxicity? So we might look at, for instance, clinical trials that are available and what are the side effects that have been reported for drugs that are purported to be used to the treatment of cancer. What do the product labels themselves say, or the package inserts? What studies have been done? So what does the literature say about it? What do we observe in electronic health records? And what has been reported through spontaneous adverse event reports? We can also look, so this is really at the clinical level, we can also look at the preclinical or even the molecular level, where we can look at the efficacy of the drug in animal models, in looking at activity of the molecule in terms of in vitro assays, so binding activity or other phenotype activities, and to look at specific targets that are known to be associated with this kind of phenotype cardiotoxicity, and so on. so we built a tool. So HyQue is a web accessible tool which you can create your hypothesis and you can, it is used then to extract the information from the various sources, present them to you, and allow you to investigate both query that was used against these resources, which are found in different databases, different SPARQL endpoints, and also begin to examine the results that are being returned. And so what's nice is it becomes a bit of a data dashboard for those data that you are specifically interested in looking at, rather than the whole of the entire set of all the data. So this is a way to customize the information that's being presented. So the next, so we have been looking at this with respect to, yeah, can you, sorry, I see that there's some messages about, okay, good. Right, so we have been examining, you know, with the evidence that we were able to draw, how well do we do against those people that have studied these drugs, or even other methods, computational methods that have made predictions about drug toxicity. And we find that HyQue does actually a really good job of correctly identifying the evidence that is available, and also being able to assess to some extent the degree of toxicity that these chemicals present. So I think that's quite promising. We've been exploring now its application in other domains, for instance, in identifying genes that are involved in aging. And again, what we see is that we can retrieve those genes that are well known to be associated with aging, but we also get the evidence for which this is true, right? So the experimental data that was used to justify the annotation that this gene is involved in aging. And what's interesting is we also find candidates that have all the promising signs of being involved, but nobody has annotated or identified them as being involved. So this is work that we've just submitted. So the next little bit that I want to talk about is some of our efforts in doing more translational research, and in particular, our efforts to link biomedical data with clinical data. And we have been trying to develop semantic clinical data warehouses that enable us to bridge the gap between these two domains. And so what's important here is that I haven't really talked about it too much, but we really see ontology as a strategy to formally represent and integrate knowledge. And it becomes almost a universal schema by which you can articulate the kinds of things that you have in your domain, but it can also be used to, when used properly in the context of automated reasoning, you can check the consistency of your knowledge. You can try to classify individuals into more specific types based on their attributes, and so on. And so we have work in those areas, but I'm going to focus a little bit on a couple of stories. One which was done in collaboration with the W3C Healthcare and Life Sciences Interest Group, which I currently chair, and for which this the Yosemite project and other health-related projects have been burgeoning from. And one of the projects that we undertook was to try to link the linked open data that we have with the biomedical ontologies. And this is kind of a challenge because there are many, many biomedical ontologies and healthcare terminologies, but there's this paucity of information to link those with the actual data that we have access to, notwithstanding the patient data. So you can see here, I have a listing of the biomedical ontologies, which include the gene ontology, the Kebi ontology for chemicals, the sequence ontology, protein ontology, and so on. And there's hundreds of these kinds of ontologies focusing on slightly different domains, sometimes overlapping in their domain, and with a set of mappings between them that is not complete, let's say, so largely incomplete and always changing with the new data that's being generated. Same with the healthcare terminologies. We have a growing number of healthcare terminologies. Thankfully, we have a few standard ones, but even the standard ones don't give you everything that you want. And so others like the disease ontology have been attempting to articulate different kinds of terms that are required for the description of diseases, for instance, and linking that then to the existing healthcare terminology so that we can use them interchangeably. So what we did was we built a translational medicine ontology, was basically an upper and mid-level ontology to connect the main concepts from the biomedical ontologies and the healthcare terminologies together so that they would link downwards to the actual data that we have in the biomedical domain, from the drug product labels, from DailyMed, drug and drug target information in drug bank, disease and gene information in OMIM, side effects and drug indications that are text mined from product labels in CIDR, information about Medicare coverage and availability of drugs, and clinical trials themselves. And so in our effort, in this very early effort, what we did was we generated RDF patient data from XML representations in the Indivo format. So we converted them in RDF. We made sure to link the concepts as well as the data items to the biomedical ontologies and downwards to the linked open data. And we linked that then to some elementary clinical decision support rules. And so what we could do is start asking questions that span biomedical and clinical research. So we could ask for, what other drugs could be used to treat a particular disease because they modulated or they acted on genes that were related to the disease, right? So in our case, we looked at Alzheimer's disease and could identify compounds that potentially could influence the dysregulated networks of disease around Alzheimer's. We could look from a clinical research perspective for drugs that have, so if you have patients that are non-responding to a particular drug, perhaps they could use a different drug because it actually uses a different mechanism of action. And so that could be a promising alternative candidate. And we could also look at healthcare applications where we could look for comorbidities, and that could influence, for instance, patient care or clinical guidelines for patient care. In our current work, now that I've come to Stanford, it's been just over a year, we have been working to transform the STRIDE clinical data warehouse at Stanford University into RDF, and to link that then with the bioretrief data sets and the biomedical ontologies we have in BioPortal. And so we have a paper that's just been accepted at AMIA, so we'll get to talk about that a little bit more. But the idea is to articulate queries across these domains and to start being able to use semantic technologies to do this translational research. So I have a few queries here, but I think at the essence here is that we can do federated SPARQL queries across our different endpoints that we have in BioRDF, and we can use that to answer questions that involve reasoning over ontologies, for instance, and the different resources that we have access to. We have been doing some research as well, translational research, where we start to use information that we have in the biomedical domain with this clinical information. In this project called Phenome Drug, what we're trying to do is identify human drug targets using animal models and their phenotypes. And so here's the basic use case where you have, if you have a mouse with a gene that is knocked out and you observe the phenotypes associated with that knockout, we hypothesize that the set of phenotypes that we observed there would be similar to if you had a drug that specifically targeted and inhibited that gene or gene product. And so in that sense, if we can match the phenotypes that we observe in humans, the side effects that are observed in using a drug in humans, we can potentially pinpoint which are the targets of those drugs. And so we do that in part by using measures of semantic similarity, where we compare human drug effects with animal model phenotypes. But this is somewhat challenged by the fact that, again, we have so many terminologies, and we need to bridge this gap. We need to make the relations between the terminologies so that we can examine their equivalence or whether or not a term is more specific or less specific than another in a different terminology. So to do our analyses, we use phenotypes, human phenotypes from OMIM, the disease database, the human disease ontology, the neurobehavioral ontology, and the mouse phenotypes. And all of these get mapped to a common ontology, which is called the mammalian phenotype ontology. On the other side, from the drug effects, we have CIDR is, what CIDR is a resource that was used text mining of the UMLS vocabularies over the drug product labels from DailyMed. And so we have UMLS terms, and we have to map those UMLS terms to the mammalian phenotype ontology. And so some of this was done automatically, and we also did some manual curation. And what this enables us to do is when we're reasoning about the different kinds of the similarity between different terms that are found in both, say, in the drug effect list and in the phenotypes from the animal model, you might have something like this. The overriding aorta, which would be expressed, say, using the human phenotype ontology, has an axiomatic description, something that we can actually reason about. And if you have an equivalent mouse phenotype, overriding aorta, we can discover that these are equivalent by virtue of their descriptions. And so this is one of the powerful things about having an ontology language like OWL, which is amenable to reasoning, automated reasoning, is you can discover concepts that are similar or equivalent based on their descriptions, rather than somebody saying this term is exactly the same as this term, but in a different organism. So in our study, we examined drug formulations, about 15,000 drug formulations, 2,200 mouse genotypes, and compared all the drug formulations with all the mouse genotypes and tried to identify drug pairs. And we did a pretty good job. So just using phenotypes alone, we can identify, we get a ROC AUC curve of 0.78 for human targets using the list that is in Drug Bank, which is pretty good. And what's really interesting is that we can begin to also understand the basis for the phenotypes that are observed by the drug, so the drug effects. So can we match the drug effects to which genes are eliciting those? And in our approach, we begin to examine, so for instance, the drug diclofenac, it's a non-steroidal anti-inflammatory agent. It's used to treat pain, osteoarthritis, and rheumatoid arthritis. And it has a bunch of different drug effects, and we can link these drug effects to this PPARG gene. And diclofenac is a known inhibitor, but about 49% of the side effects are explained by the knockout, the mouse knockout. And we can also, it also targets COX-2, and again, 46% of the side effects that we observe can be explained by targeting this particular gene. So overall, the aim of my research is to understand how living systems respond to chemical agents, and by developing applications for these drugs in small molecules, for instance, by looking at drug repurposing for rare, complex, and untreatable diseases, looking at different ways that we can combine drugs and try to minimize side effects, looking at the role of metabolic products in toxicology, which is relatively unstudied, and also figuring out how we can use small molecules in synthetic biology and sort of drive the next generation of applications, of biotechnological applications. So in this, I think one of the issues that we have is that it's relatively difficult to get access to patient data. This is one of the barriers of research. I'm fortunate that it's relatively easy at Stanford, largely because they have a program to prepare clinical data, so again, the STRIDE clinical data warehouse, and also to provide us with de-identified patient data. And because of that, it's relatively easy at Stanford anyways to get IRB approval for our research because it doesn't contain any personally identifiable information. Now, if the organization that you work with doesn't have support in this kind, it can be really difficult to extract the information from, for instance, from an EPIC installation or Cerner and to prepare it and to go through the whole procedure, and it's a huge barrier. So we're hopeful that as deployments of clinical data warehouse technologies become more prevalent and easier to apply over existing health data information systems, that more people will be able to make use of the kinds of approaches that I've discussed in order to transform data in a way that can be easily interlinked and queried. And a big point on this is that for the SPRIDE database, they made enormous strides to use the standardized healthcare terminologies like SNOMED, ICD, RxNorm, LOINC, and so on. And this is a huge step. Now, while those terminologies themselves have deficiencies, it's still better that they use a standardized terminology to the extent that they can, and then we try to augment above and beyond that using the other linked terminologies. We and others here at Stanford and around the world are starting to use text mining over the clinical notes and trying to make sense of those textual data, which are actually far richer than the structured data elements that we see in the AHR. But of course, with the increase in information is also an increase in the noise. So there are lots of sort of false associations that are there, but they can be weeded out if and only if the kinds of studies you're doing involve large numbers of patients or observations, and you're not asking questions about specific individuals. In the TMO work that I discussed, we were trying to answer questions about individuals, and so this requires a certain level of precision or at least some ability to mitigate the potential for false positives associated with, say, text mining. We're also focusing a little bit of our attention to try to better integrate the terminologies that are used in the biomedical domain with the clinical domain. And so it's been encouraging that just this past summer, the human phenotype ontology, which we strongly depended on for our study along with the mammalian phenotype ontology, is going to be incorporated as part of the UMOS. And so that makes it vastly easier to move from, for instance, the human phenotype ontology, which contains at least 50% of the terms that it contains are not in the UMOS, but are useful in both text mining, these clinical notes, and also for mapping to animal model phenotypes. So we're encouraged by, I guess, organizational efforts to link these two together and produce standard distributions that everybody can make use of. Also, I think, in general, we are developing and we have access to a growing suite of methods to access and integrate data using these semantic technologies. So again, RDF as a common base level platform for representing data also enables us to transform these data to, for instance, meet particular standards. So if you had, we have seen discussions previously in this series about FHIR and having RDF representations of healthcare data. Well, you can, with RDF itself and with SPARQL, you can easily syntactically transform our existing RDF data into a new common format. And I think that's really kind of promising, that everything can be done basically within the framework. You don't have to go very far outside of that. We use OWL ontologies not only to tag the data themselves, but also as a way to formalize the meaning of terms and to ensure that the description of these terms are sufficient so that you can check the consistency of those terms that you're using and in the context of the data that you have. And I think that's a growing need and it has yet to be fully explored. And we and others are developing methods to integrate, to query, and to semantically compare these data. And I think as they become more mainstream, they will enable many more applications. So for instance, Peter Robinson and others are examining how can we use animal models to also try to diagnose patients with rare diseases. And I think that's kind of interesting, innovative application of semantic descriptions and semantic technology to for clinical and for clinical research and for the delivery of healthcare. So we are basically at the start of something pretty exciting, that if you can get the data that you're interested in and become familiar with these methods that we're developing, we and others are developing, that there's enormous potential for applying them for testing, diagnosis, treatment, and many more applications that we have yet to envision. All right, so with that, I just wanted to thank some people, particular in my lab. So Jose, who I think is on the line, is now currently at Bioinformatics and has been instrumental in helping develop Bio30F. Same with Allison Callahan, who has been working on HyQue and is currently a postdoctoral fellow in Nigam Shah's lab here at Stanford, just down the hall. Tanya and Beatrice were working on the TKI Cardiotoxicity project for the FDA. I should mention that we're looking for postdoctoral fellows, and so if you know somebody or you are looking for something, please let me know. I also want to thank our numerous collaborators, those that have contributed to the Bio30F open source project, all the participants of the W3HELS, especially those that were involved in the TMO work and other pharmacogenomic work that I haven't mentioned. Mark Wilkinson, who has been developing the SADI Semantic Web technology, which we have been applying to Bio30F and to use in the healthcare domain. Robert Hundorf, who's now at KAUST, who has been instrumental in the the phenome drug and other investigations with semantic similarity. George Kutos, who has been involved in the development of terminologies, such as the phenotype ontology, PATO, and others. And Nigam Shah, who has been involved in many conversations, particularly about HyQue and Bio30F and so on. Finally, I want to thank the other members of and drivers of the Yosemite project, and also Eric from SemanticWeb.com, and all of you for taking the time to see this presentation, and hopefully I can now take some questions and that you may have, some burning questions that you may have, to focus our conversation about how we can use semantic technology to link, I think, biomedical data with healthcare data and really do some cool, exciting things. All right, thank you very much.
Michel, wonderful. Thank you so much. We are now at the Q&A portion of today's webinar, and to kick us off, David Booth will actually be posing the first question to our audience. David.
Yeah, so I want to turn it around and ask the audience a question to think about, and maybe you could please type your suggestions into the Q&A window of the WebEx interface. And the question is, if we were to arrange some kind of additional webinars or other follow-up to this webinar series on the Yosemite project, what topics related to the Yosemite project would people like us to cover? So if the audience could think about that as we're doing the Q&A and just type your suggestions into the Q&A, that would be great. Okay, and, Eric, you want to? Yep, go ahead.
Sure. As a housekeeping bit, to keep things a little bit easier on my tired brain, if you would please flag those suggestions with the word topic at the beginning. Say topic, colon, or dash, and then type the topic you'd be interested in hearing. That way I'll be able to differentiate them from the questions quickly and easily. So to kick us off on the questions that people have been submitting, we're going to start with a couple of workflow tool questions. So the first one, Michel, is what conversion scripts from GitHub get used to convert the original data to RDF? Is that published anywhere?
Yeah, so the project is open source, and the source code is available at GitHub. So I pasted in the link as an answer to that question. We also have a paper that we have that was published as part of BSWC that describes that release. I think it was release 2, and we're now at release 3, and so we're just preparing a paper to describe that effort too.
And for anybody on the listening to the recording later, the link that Michel pasted in was github.com/bio2 RDF/bio2 RDF-scripts.
and we will post that alongside the recording as well. Okay, so the next question is, given the high complexity of interlinking and applying all of those different ontologies to the data, I'm assuming this person is referring to the slide where you showed the various ontologies you were working with. So given the complexity of working with all of those, what ontology tools do you use to accomplish this?
Well, that's a good question. From the tooling effort, I mean, if we're using ontologies, we're typically using the OWL API that facilitates the parsing of ontologies and also their modification or the creation of new axioms, at least programmatically speaking. So when we have data that we can formalize using a common pattern, we can typically do it programmatically fairly simply. And so we have some papers where we show how to do that, where we integrate drug bank, pharm GKB, and CTD together along with the terminologies, the 7 terminologies, and build a coherent sort of integrated knowledge base. So we can do that programmatically. The challenge, I think, more is that when the mappings themselves between the different terminologies are poor or could be improved, how do we do that? And I have to say that thus far, I've been less than satisfied with the tools that are existing to facilitate mapping efforts, although I do know that there are some commercial tools to help you do that. And so the big part of the challenge is to have the appropriate expertise to find common matches. And so, for instance, in the TMO work where we did map many of the biomedical and healthcare terminologies to the translational medicine ontology, it was largely a manual effort where we searched bioportal for terms from the different terminologies, and then we manually created assertions talking about whether or not these terms, these concepts were equivalent, or whether they were one would subsume the other or whatnot. So I would say if it's pretty straightforward and you're going to apply a uniform application, then we use programmatic access, usually with the OWL API. In other words, we tend to create spreadsheets where we're going to curate information about the mappings, for instance, and then we might formalize that with a little script of some kind.
And this is David. I just want to add, so in the Yosemite project roadmap, in step 4, crowdsourcing translation rules, the reason for approaching that in an agnostic way about how those translation rules are written is that this area is still fairly immature, as Michel kind of alluded, that there are a variety of ways that it's handled, and it's not really very clear what ways are going to be really the winners. So that's one of the reasons why we're agnostic about that in the step 4 in the roadmap.
The next question, how do you handle cases where similar language suggests equivalence, yet there may be danger associated with calling them equivalent?
Yeah, so this is a good question. I think people often get held up on, the semantics and the precision that is required. And I would basically say that probably from an information systems perspective, you would never actually assert equivalence, but you use some vocabulary that you could then interpret in a second way. So you just want to keep track of the relations as you find them, as you believe them to be. But when it comes to the application, the thing you're actually trying to do, you may apply a stronger semantic than most people would be comfortable with. And we often do that. We make decisions about what the terms and their mappings mean, and we use that to our advantage. And so that means that maybe these mappings would not be universally agreed upon, but it doesn't really matter because it's in the context of a specific application where applying those semantics is exactly what you need to do. So my suggestion is curate to the degree of accuracy that you can, and then in your application, formalize it to the need that you have.
Okay, thank you. The next question, how do you see semantic discovery services like SADI being used by healthcare professionals or even patients? Ideas about applications that can or will be constructed for this end? And I may ask David to jump in on this one as well. So go ahead, Michel.
Sorry, I was just looking for the question. Is it in the Q&A?
Yeah, it's down at the bottom. You may need to scroll down.
Yes. Yeah.
Oh, okay. Oh, so how for SADI? Oh, I see. Okay. Hi, Bob. So this, with Mark Wilkinson, we actually published a paper where we were, let me see if I can remember exactly. This was with Mark Wilkinson, and what we were trying to do is empower patients to understand potential drug interactions, drug-drug interactions, given their own EHR data. So we did this demo where the patient had access to their own patient record. We knew a list of drugs that they were taking. And of course, patients browse the internet all the time because they might be suffering from side effects, or maybe the drug isn't very effective. So they look on forums to see what other people say about the drugs, or what other options they maybe could consider. And so what we tried to do there is to use the SADI framework to basically look up for the drugs that they have any known interactions from other databases and present that on the page that they were browsing to say, yeah, there's this drug and you might be considering it, but there's known drug-drug interactions with the drugs you're already taking. And so that's an example, I think, fairly straightforward of how we can use semantic web services to access data on demand and integrate that into the user experience. Currently, we did it that's a patient-centered case, but for the physician, I think some of the conversations that we're having here at Stanford is to see how can we leverage the fact that there have been patients before the one that they are seeing their history and their outcomes could inform what to do with the patient that you're looking at right now. And so if you can find cohorts that are similar to the cohorts that are similar to the patient you are looking at, then maybe you can prescribe a better treatment plan than you would have otherwise. So I think the SADI Semantic Web Service Framework is a way to provide standardized APIs to data that might be on the back end. Well, it could be any kind of data, but for us, it's a sort of discovery, service discovery, and a platform.
Great. For our next question, we turn to the business benefits. In the future, how do you see semantic data projects like this affecting the cost and effort associated with researching and developing drugs, and how so?
That's a really good question. We are currently working with different pharmaceutical companies, and I think one of the things that has always struck me is how amazingly disorganized from an information perspective any company really is. And I think one of the nice things about semantic technologies is that we can sit down and not have to change necessarily their business process, how they currently do things, but we can strategically add value where it makes sense to do that. So for instance, if an organization has multiple sites and multiple databases in different locations, connecting these up in a way that's, that makes sense is something I think that semantic technologies are well suited to do. So instead of embarking on a massive, let's merge these data sort of enterprise and let's change our business process, I think just by coming in and saying what's the real goal that you have and focusing on that and building a project on top of existing infrastructure is a way to save money from otherwise difficult projects.
And this is David. I'd also like to add that there's also interest in pseudo-randomized clinical trials, where you look at existing clinical data and just observing what the effects were, what the outcomes were, and controlling for lots of variables in a well-defined way. And instead of actually conducting a clinical trial, you sort of pretend that you did a clinical trial using that existing data and controlling for the various variables. And so that it can be a far cheaper way to at least reduce the number of candidates of drugs, for example, that need to be explored than as is currently done.
Well, thank you.
I think that's it.
Yeah.
Now, before we finish up, I also just want to mention again, if we were to arrange some kind of additional webinars or follow-up, what topics related to the Yosemite project would people like us to cover? So if anybody has any other thoughts on that, please put them in the Q&A.
And if if something hits you in the next 24 hours or the next week or in the near future, feel free to send those topic ideas directly to me. That's Eric, E-R-I-C, at SemanticWeb.com. And I will collect those, and David and I will continue talking about that. So thank you very much to Michel Dumontierl today. Thank you to David Booth, and thank you to all of you. This has been a really great presentation, a wonderful series on the Yosemite project. I know I have learned a great deal about this effort, and there's a lot of exciting stuff going on. If you missed any of the episodes, they are available in the media section of SemanticWeb.com under recorded webinars, so you can look for those there. Thank you, all of you, for your time and for tuning in today. Just to remind everyone, we will be posting the recorded webinar and the slides to SemanticWeb.com within 2 business days, and you will be receiving a follow-up email to let you know how to access that material. That will go out to all registrants. So thank you very much.
Thank you, and have a great holiday.
Happy holidays.